This video is on protein synthesis. It is part of the Gene Expression Achievement Standard, and synthesis means creation, and so it's the process on how proteins are created. The following PowerPoint is at an achieved level standard. Right. There are two parts of protein synthesis. The first part of protein synthesis is transcription, and this occurs in the nucleus of the cell, and it makes a strand of mRNA as its product. So that's the end result. That's what you get at the end of transcription. Second part of protein synthesis is translation. That occurs in the cytoplasm, and the end result of that process is that you end up with an amino acid chain, which will then fold in to make the protein that was instructed by transcription. This is the overall, we'll do transcription first. This is what the whole process looks like, just to give you an idea. So the red here is the mRNA that's being created. The mRNA is only one strand, whereas the DNA is two-stranded. So the DNA doesn't leave the nucleus because it's double-stranded, whereas the mRNA still has the instructions of the DNA, but it's only single-stranded, so it can leave the nucleus because it's skinny enough. Let's do it in steps. Number one, so a chromosome is made up of many genes, and in protein synthesis, one gene unzips. So, step one gene will unzip. An enzyme called polymerase attaches to the start of the gene, and the start of the gene is called the promoter. Second step, the polymerase, the enzyme, moves along the template strand, which is the strand here at the bottom. And it will start to build the mRNA strand by using the complementary base pair rule. But instead of thymine, it will use uracil. So if we have a look at the mRNA strand here, these bases are complementary to these bases on the template, but instead of T, we use U. These nucleotides, so these bases are just floating around in the nucleus all the time. Third and last step, the polymerase will then reach the end of the gene, and the end of the gene is called the terminator region. The mRNA will then detach, and then it will go through little pores of the nucleus into the cytoplasm, and that's transcription. That gene of DNA will now zip up again, and it will re-roll into the helix shape. Now we're on to translation. So we're in the cytoplasm now, and this is our, I'll take you back. This is our mRNA at the bottom. This blue thing is called the ribosome, and the ribosome attaches around the mRNA. Don't worry about this writing. And the ribosome then attaches these amino acids that are being brought in by these things here, which are called tRNA. And when we get to the end, we'll stop codon. The polypeptide chain then falls off and it folds into the protein. So, steps number one, the ribosome, which is this big purple thing here, will attach to the start codon on the mRNA. The start codon is always AUG. The ribosome's job is to read the codon sequence. So a codon is three bases together. So AUG, CUC, AGC, GGA, UAA. The ribosome is to read the codon sequence and then bond all of the amino acids together according to that sequence. So, step two. Molecules of tRNA bring the amino acids to the ribosomes in the correct sequence according to the mRNA. Each tRNA molecule has an anticodon, and the anticodon are these three bases here. an anticodon is complementary to a codon for an amino acid. So if we see here, this is the codon, CUC, and the anticodon is GAG. So the tRNA will come in. The next one is codon is AGC. So the anticodon is UCG. So an anticodon is complementary to the codon for amino acid, e.g. the amino acid lysine has the codon AAG, so the anticodon is UUC. Therefore, lysine would be carried by a tRNA molecule with an anticodon of UUC. So as the tRNA molecules come in, they leave the amino acids behind, and polypeptide bonds form between the amino acids as they're brought one by one to the ribosome by the tRNA. This creates a poly, which means many, polypeptide chain. The chain of amino acids keeps growing until the stop codon, and in this case the stop codon is UAA, is reached, and there's no amino acid for the stop codon, so then the chain will just drop off, then it will fold into the protein. Okay, so hopefully that was clear, and if you write down the notes from the PowerPoint, then that should help you with your study as well.